In the first model, we assume that the ligand activates the receptor and that we have a reversible deactivation. In the second model, there is no reversible step, but we have an irreversible internalization of the receptor. And the third model comprises both processes. Andrea Pfeiffer, also from the lab in Heidelberg, from Ursula Klingbeller, measured two experiments, one with continuous stimulation of the system, and the other one with 5 minutes pulse. She washed away the ligand after 5 minutes, which was possible because the cells are adherent to the dish. Now in the upper panel of models, you see that each model is coupled to one of the two data sets. And first we are going to fit model 1 to the continuous stimulation. So in green we see the constant ligand concentration over time, in blue the data points and the arrow bars, and in red the model. Well, this fit doesn't look very well, so it seems that model 1 is not able to explain the data. It can, however, explain the pulse stimulation. We go ahead to the second model, and this model is apparently able to fit the continuous stimulation and also the pulse stimulation, but we did this only separately. We want to have the same parameter values actually for both experiments, so we have to fit both data sets at the same time. If we do this, then we realize that this model is not able to capture the dynamics of both experiments simultaneously, and we can rule out this model too. If we go ahead to the third model and apply right away the multi-experiment fit, we see that it's not yet perfect, but still significantly better than before. The ligand concentration is here used as a stepwise analytical function. We can also use experimental data. In the second model, for the JAK-STEP pathway, we assume that the phosphoreceptor activates the start molecule. We have dimerization, the dimer enters the nucleus, and in this model there's no flux out of the nucleus. I cannot see this on the screen in Freiburg actually. I think that we have some delay here maybe. Can you see the JAK-STEP pathway? Yeah, I think I have no transmission from Freiburg back to Boston. I just continue. If we I think you don't see any changes on my screen. Is this correct? I cannot hear you. Okay, then I have to continue without a picture, which is
Thomas, just go ahead, Thomas.
Okay. So if we try to fit the data for the JAK-STEP pathway, we realize that the model with the route and the flux of nucleus we are not correct. And if we go ahead to the second model, we introduce the change. Sorry, this really makes no sense. What is going to show you this on the screen? I will go back to PowerPoint and hope that you have the screen again. Don't have this either. Yeah, so I will just conclude my talk. Could you, Mr. could you my PowerPoint presentation? And this takes too long, I guess. So let me just summarize without some slides. The Prozesses software has evolved since its academic state into a professional software, and you can just download it for free for academic usage from Prozesses.de. And it's very nice to have this close collaboration with the people in Heidelberg, which was required for this interdisciplinary research field, and to implement all our experiences so that other users can benefit too. Let me thank again the organizers, and I can speak to you via this web conferencing system, which in all tests before worked quite well. And the funding by Hypothesis and the Crossbase Foundation. Thank you for your attention.
Thanks a lot for this very nice presentation, and I think we got the major points despite the technical problems. I have a question regarding Potter's wheel. You explained that you can compare the different models, and one of the model explains the data and the other one doesn't explain the data that nicely. Are there other measures to compare the models more objectively that you could suggest to use? For example, like the Acker-Ike information criterion or so? Is that implemented?
Sure. So, if we apply a chi-square test, I mean, now we just visually whether the model explains the data, yes or no. But if you have realistic values for the app, which is very important, then you can calculate a statistical measure, a p-value, that tells you that the model is statistically sufficient to explain the data. And if you have several models, you can compare either the Acker-Ike criterion or you can compare nested models using a live iteration test.
Are there further questions?
Today we discussed some observations on heterogeneity of populations, and this may also hold true for some of the experiments which you have your hands on. How would you like to solve a proof or a counterproof of heterogeneous cellular reactions within a cell population under study?
Yeah, it's a very important question. During this multi-experiment fitting, we distinguish between two kinds of parameters. One kind has the same value for all experiments, these are global parameters, and the other parameters may have different values for each patient, for each cell, for each experimental condition, these are local parameters. And this way we can deal with both situations.
So if there are no further questions, then let's thank the speaker again.